First, in a series of videos, we're going to be looking at the fantastic mechanisms of the immune system. In the course of these videos, we're going to look at components and targets of the immune system. We're going to look at innate immunity and acquired immunity. And with innate immunity, we're going to focus on inflammation, interferons, natural killer cells, and the complement system. With acquired, also called adaptive immunity, we're going to focus on B lymphocytes, which are involved in antibody-mediated immunity. And we're going to look at T lymphocytes, which are involved in cell-mediated immunity. We're also going to focus on immune diseases. Shown here is a human immunodeficiency virus, which you see here in gray. This is the virus that causes AIDS, and it's attacking a helper T cell or T lymphocyte, which is HIV's primary target. This is some computer artwork showing a macrophage with these extensions coming out and grabbing onto a virus. And what you see here in green are the antibodies that are attached to that virus that are alerting the macrophage to phagocytosis virus. This is a scanning electron micrograph showing the size of a red blood cell compared to a platelet and then compared to a T lymphocyte. So in dealing with immunity, this is the body's ability to protect itself from its own defective cells, as well as from bacteria and viruses and other pathogens and toxins. Here we have a young child who's kissing a pig, and don't worry because that young child has a fantastic immune system to protect it from any type of microorganisms that come across and possibly infect that child. The pig died a few days later, but the child is fine. It just shows that we come into contact with a lot of different pathogens, and yet our body can handle most of these pathogens and protect us. There are three major functions of the immune system. The first is it's going to remove damaged and dead body cells. Shown here is a macrophage that's gobbling up red blood cells. These are old red blood cells. Remember, they have a lifespan of about 120 days, and then the old ones are going to be broken apart and destroyed by cells such as the macrophages. Number two, it helps to remove abnormal cells such as cancer cells. We're going to see that the immune system plays a pivotal role in removing cells that become cancerous from your body. The third is that it's going to protect you against invading pathogens, such as bacteria and viruses and fungi, protozoans, and multicellular parasites. For example, fungal infections such as ringworm. Ringworm sounds like it's an infection by a worm, but it's actually a fungus that's infecting the body. Well, in looking at bacteria, these cause tissue damage and disease by releasing enzymes and or toxins that are going to disrupt cell organ structure and function. Bacteria have a plasma membrane. So shown here is a bacteria. This is going to be the plasma membrane here. And then inside of that bacteria, you have cytoplasm. And DNA is the genetic material of bacteria. So they have their own cell machinery for replication and metabolism. We're going to compare that to viruses, which do not have their own cellular machinery for replication and metabolism. And bacteria can be killed by antibiotics. Well, most bacteria have a cell wall that surrounds them, and some can have a capsule that protects them against the immune system. Viruses, these are not cells. These are not technically alive cells. They're what's known as obligate intracellular parasites, meaning that they only carry out metabolism and reproduce when they're in a host cell. So a virus has to enter a cell, and then with its genetic material, it's going to basically take over the cell's machinery to make more viruses, but it can't replicate on its own. Viruses can be classified based on one, their type of genetic material. Viruses will either carry DNA or RNA, but don't carry both. And two, their structure, such as being enveloped or non-enveloped. And shown here are examples of enveloped viruses. They have a phospholipid bilayer around them, contrasted with non-enveloped viruses, which don't have this membrane envelope around them. You want to make sure you star this. Retroviruses have the genetic material known as RNA, or ribonucleic acid. For example, HIV, which is a virus that causes AIDS, is what's known as a retrovirus because it has RNA. So retroviruses contain RNA. And although we're not going to go into the details of how retroviruses work, They're really fascinating. When you take microbiology, you'll go into these type of details. But basically, when you look at a retrovirus, like the HIV virus, this infects the cell, and within the coding of this HIV virus, you're going to have RNA. And there are these enzymes that it carries called reverse transcriptase, which will take that viral RNA and reverse transcript it to DNA. And then that DNA can insert into the DNA of the host cell and then make RNA from that, which is going to direct the assembly for more of these retroviruses. It's a fascinating process by which they can reproduce. Now, the structure of all viruses includes a protein shell called a capsid, and that can either be enveloped or non-enveloped, also known as a naked virus. Shown here are two viruses. This is a capsid, and both viruses contain a capsid. It's that protein coat. Well, if you're a naked virus or non-enveloped virus, you don't have a phospholipid bilayer around it. If you're an enveloped virus, then you do have a phospholipid bilayer around that capsid. This enveloped virus has this additional membrane layer that covers the capsid. This is a layer that came from the host membrane. So basically it stole it from the host membrane when it budded off. So here's a virus, and it enters into the cell. Its RNA directs the cell to make more of the virus, as well as the different proteins that are going to find on that plasma membrane of the enveloped virus. And then it puts these proteins on its cell surface. So whenever the virus leaves by budding and it takes some of that cell membrane with it, those proteins were directed by the virus to be made by that host cell. It gets put on the membrane, and then when the virus buds off, then it has these proteins on it. For example, the COVID-19 virus, the proteins are part of this membrane envelope that it took from the cell when it came out of the cell. So in addition to the host cell proteins, it also has the viral proteins that are directed the cell to make. And these bumps and knobs and spikes that artists use in images of enveloped viruses, like for example in that COVID-19 virus, this depicts the structures on the viral envelope that we see here and here, as well as here. And this image shows a little bit more detail about how a virus infects a cell, directs the machinery of the cell to make more viruses, as well as the proteins that are going to be on the enveloped virus, and it puts it on the cell surface. So when the newly made viruses bud off, they have an envelope around those with those proteins that it directed the cell to make. Now, when you get to microbiology, you'll go through all the details on how this happens. This is beyond what we're covering in physiology, but just to get an idea what an enveloped versus non-enveloped virus is what you need to know. Common disinfectants and even alcohol, detergents, or soap can disrupt that oily envelope and its components, which destroys the ability for the virus to infect host cells. We're going to look at, for example, the COVID-19 virus and these spike proteins, which are used to bind onto cell receptors and be pulled into a cell that's in your body. These substances, such as disinfectants, can disrupt this membrane around here and prevent the virus from invading your cells. Examples of enveloped viruses includes ones that cause the notorious diseases in humans such as SARS-CoV-2 that causes COVID-19. We have the influenza virus, hepatitis B and C, hemorrhagic fever, or the Ebola virus. And these are examples that you don't need to memorize, but just give you an idea of the type of viruses that have these envelopes. Now, non-enveloped viruses do not have a lipid covering and have just the capsid. And these so-called naked viruses only need their protein-based capsid and host detector proteins to infect a cell and bind onto that cell and enter the cell. However, because they lack a membrane envelope, they're more resistant to many disinfectants and other stresses like drying out or heat. Now, examples of these non-enveloped viruses include types that cause dysentery, common colds, and poliovirus. Virus replication occurs inside a cell, and depending on the type of virus and its outer coat properties, it can enter the host cell in different ways, the details of which are going to be covered in microbiology. But once it enters that host cell, it's going to induce that host cell to make more of the virus, and then the virus is going to be released. The release of the virus in the host cell can occur by lysis, where the host cell basically explodes with new virus particles spilling out. It can occur by exocytosis, the new viruses. It can occur by budding, where the enveloped virus particles surround themselves with a layer of the host membrane and then bud off from the surface of the host cell, as shown here. Now, viruses can cause cellular damage and death by a number of different ways. You don't need to memorize all these different ways that viruses can cause damage and death, but this gives you an idea. They can lyse the cell upon release. They can cause the cell to produce toxic substances. They can disrupt the cell's metabolism, resulting in death of the cell. They can induce the destruction of the cells by the body's own immune system because the infected cell is no longer recognized as your normal body cell. Some can transform the cell into cancer cells. Some viruses, such as herpes simplex type 1 and the varicella zoster virus, which causes cold sores and chickenpox, respectively, these hide out in cells and then they can replicate sporadically. Some viruses incorporate their genetic material into the host cell DNA. And viruses with this characteristic, which include HIV and oncogenic viruses, these can cause cancer. And shown here, this is a virus-infected cell that's put its viral proteins on the cell surface. And then what you're going to see is T cells coming in, and these T cells will recognize these foreign antigens on the surface of the cell, and then release chemicals that cause the cell to die by apoptosis, thereby killing the cell to protect the body against, the virus-infected cell. Well, there are also inappropriate immune responses. So, for example, allergies are a response to normal, harmless substances. You have autoimmune diseases. This is where the immune system attacks a person's own body. And you also have a lack of immune response, such as in immunodeficiency diseases, which arise when some component of the immune system fails. All right, let's test your knowledge. Part 1. Go ahead and pause your video and answer the following questions. Here are the answers. Here's part 2. Go ahead and pause your video and answer the following questions. Here are the answers. Here's part 3. Pause the video and answer the question. Here are the answers. This concludes the video, and if you have any questions, let me know.